Wow, thank you. Hard act to follow, but thank you very much for letting me talk. Let's see, how do I go? All right. Oh, are people doing the things? All right. Okay, thank you so much. So I'd like to talk about some ideas. Now, is anybody else in the room not a biologist? It's just, oh, thank you. Oh, good. Because I feel terrifically biologically challenged. And I want to say everything I know about biology I've learned from Marianne, Anita, and a couple of other people over the last period of time. So thank you so much for a brief education. My own background is in physics. So many of the things I say might sound very naive. But I'd like to share with you a little bit of my understanding of biology and some ideas about how we can communicate. Also, thanks to people at CMU, Ed Hovey, Gully Burns, Tim Clark very much should be on there as well, and Phil, for communications we've had in the past and that are continuing at this workshop. All right, so this is what I understand about biology. That actually, I won't go over all of this, the slides are online, but specimens are not species. So all different animals in a certain species are actually really, really different. They have different amounts of venom, or they're not even, it's not even clear what strain or what they belong to. And then gene expression varies hugely. Anita helped me figure out some of these examples. So with age and smell and hunger and light, with all of these things varying, all of these different ways in which living systems move through the world, their gene expression, the things that they're making from their genes constantly are changing. So knowing genes is not knowing how they're expressed. No man or mouse is an island, and I found this absolutely fascinating to read about all the microbiomes. They start to colonize an infant when they're born, and they're very, very diverse, and they're very different per person, and there are 10 times more of them than you have cells in your body, which is just fascinating. And then interactions create more complexity, and I had found this quote before I realized that James Glazer was talking here. But I really like this quote in the context of this workshop. No amount of information about what happens inside a single cell can ever tell you what a tissue is going to do. So even if you have the perfect 3D virtual cell, there is so much information that is going to be needed if you're going to be able to, if you want to try to figure out how that cell is working within a body, let alone within an animal, let alone within a species. So these are all huge problems in biology. And what I've learned at this conference even more is that models and experiments are very, very different. You're not modeling what the experimentalists are measuring directly, and if you are, you don't know because you're talking in different languages. And then none of this is in equilibrium, so in fact it's all constantly changing. So in other words, the main thing I've learned is that you cannot think of living systems as automatons. This is a drawing from the 18th century, which actually was used to illustrate the fact that animals are machines and apparently people are not. But it really doesn't work that way. If you take an animal apart into its constituent components, you can't put it back together and get it to run again, right? So life is really complicated. And in other words, the whole reductionist paradigm doesn't work for living systems. You can't take it apart and put it back together and expect that you know now how it works. So one thing that, say, other sciences do at this point is that you say statistics to the rescue. And you can find a number of papers in science where if you have enough populations, you have enough samples, you have enough experiments that you can actually say some things. So here's some quotes from the Human Microbiome Project and a paper recently in Nature News where people say, well, if we have enough samples, we can actually say certain things, right? If all these things are so different, then surely collecting a bunch of statistics would really, really help. And so the idea that might really help biology would be to have incidental collaboratories. So the term collaboratory, does everybody know that? Anybody not know it? Okay, all right, so the idea is that you work together with another lab. You don't create your own data and analyze it yourself, but you use somebody else's data. Most of the collaboratories have been not incidental, they've been on purpose, but you can enable the creation of incidental collaboratories where you would store data at the level of the experiment, accessible through a single interface with metadata that allows you to know what was done and what was seen. You then connect it so that you would allow analyses over similar experiments types and experiments on similar biological things. And I added here in a way that might be able to be used by modelers, right? And then you want to keep it. You'd like to keep this stuff around for a while. You'd like to probably fulfill your data management plan requirements, but actually they mean something because this is how science is expressed. So you want it to live after you are no longer doing that experiment so people can use it in the future, and probably want some gated access, and I'll get to that in a second. One of the problems, however, in doing this is what I've learned is that biological research in particular is quite insular. So there are groups that prepare things, they observe things, they analyze, they think about them, and they communicate about them with the outside world, but they keep everything that happens inside their lab. And I understand this goes mostly for wet labs, but it apparently also happens in computational biology a bit. And I was wondering why this is rather different perhaps than some of the other sciences. One of the things perhaps is that biology is quite small, so the size of the objects that you're working on is between maybe a couple of microns to a couple of meters. It's something you can hold in the palm of your hand, which is not the case, say, if you're working on a galaxy or you're working on a mountain. So you can work on it by yourself. And then it's messy. You need to actually deal with bodily fluids and such, and chemicals in which that are not always pretty. So you can't be sitting behind a terminal to do this. It's competitive. And in other words, I think the structure of biological research allows people to live in a rather insular way compared to some other sciences. So there's not this innate collaboration. So if you look at a typical lab, the question is, how do you get the right antibody IDs? So we're also working with NIF. I think it's a requirement to be up here that you work with NIF, but they're just really good at setting up collaborations. But how do you get the right IDs for the antibodies, which are injected in very messy environments? From the lab notebook, and this is a picture I took 2 weeks ago, this is the state of the art of record keeping in many, many labs, into the command center of a PI. So the PIs do want to have an overview of what's going on, but all the things are happening in a different place, in a messy way, on paper, etc. And so we've heard a lot of objections and rebuttals regarding data sharing. Our lab notebooks are on paper. So the rebuttals are really things that my group and I are interested in doing and interested in working with people to try to do. Perhaps we can develop some smartphone and tablet apps and work with existing lab software to get some of the data in at the point that it's being measured. I need to see a direct benefit from something that I'm spending my time on, so the question is, perhaps we can develop data manipulation dashboards so that the PI can see exactly what's going on, because if it's all stored in paper notebooks, they're not able to do that right now. I want things to be peer-reviewed. So the concept would be that you have a gated access to this experimental repository that would allow certain people to see your data and not others. And I don't really trust anyone else's data, so I don't want an experimental repository that I can look at. Well, maybe a couple of people I would trust their data. So you'd need there to be a social networking component. But perhaps the biggest hurdle that we've heard is that people are afraid that others might scoop their discoveries. So I think one of the key things, if we want to move towards a world where experiments are better connected, is to have a reward system that moves from a competition to a shared mission. And actually, this is where I hope this conference might be very, very useful if you can define such a shared mission. So very quickly, the idea is that you would enable collaboratories. So you kind of pop open this lab bubble. You allow the observations to be stored in a certain way. Your labs are no longer information islands, but they're more like sensors in a network. And this term came from Marianne. Conglomeration of evidence can happen. It would be a place to allow you to share negative data, and the whole reproducibility concept would also work here. It would also allow you to do joint experiments, and I was thinking about the stories of antibodies that are essentially shipped all over the planet. You might think of it that these antibodies are they're subjected to all kinds of experiments, so wouldn't it be interesting if you could track them, compare the outcomes of the interactions they have had, so that you can make what I'm thinking of as a physicist essentially like a virtual reagent spectrogram. You know, you send these things all over the planet, you see how they interact, then you pool all the ways that they have interacted. And this can tell you a lot about the structure of these entities, these actual things. This could be chemicals, it could be antibodies, it could be lots of different things. It could be strains of mice. So that you actually connect what has been done with these things, right? So within the scope of FORCE, we've been looking at an environment, if you have a workflow-based environment where you add metadata at the point of doing the measurement and then more or less graft a paper on top of this. I think this also connects to Tim's idea of store and add metadata and record where your experiment happens as it happens, and then write a brief narrative surrounding it that you share. Oh, I gotta stop. All right, very quickly, this is my department. We want to increase the amount of data, the value of the data, and deliver credit. We want to build tablets for Carnegie Mellon and link to local software to connect these runs. And in summary, I think it would be great if we are able to create such a connected experimental platform. And I think the social barriers are the hardest to cross, and maybe this workshop can define a common mission for that. Thank you.